Hello and welcome. Let's make a eyepiece graticule today. If I show you a diagram of a frog and ask you to guess its size, you'll easily think of something like the size of your palm, or maybe about as big as a potato. When we look at objects we see every day, it's easy to imagine their relative size. But when it comes to microscopic structures like the cilia lining the human gut, it's much harder. Just looking at a diagram doesn't help you feel how small they truly are. So let's try something. On your screen, you'll see a list of biological structures. Pause the video and try to arrange them from smallest to largest. Let's see how close you got. The width of DNA packed tightly inside a cell's nucleus is the tiniest, and the early stage human embryo is easily the largest. You might have placed bacteria and mitochondria, as well as yeast and red blood cells, a little differently, and that's okay because their sizes often overlap slightly. But what's truly fascinating is the difference in the scale. For instance, a virus can be up to 100 times smaller than a typical animal cell. That's the beauty and the challenge of exploring the microscopic world. Let's look at this fluorescent microscope image of a cell. The cell glows green and cilium is red. The cilium is zoomed up in the inset. Can you guess its length? If you said about 1/3 of the length of the cell, you're on the right track. It's likely around 3 to 10 micrometers long. Let's add a scale bar now. Look at the cell and not the zoomed up inset. What do you think? 4 micrometers? You're developing an eye for microscopic measurements, and that's great. Now in this video, we'll take that one step further. We'll learn how to measure microscopic objects using just a few simple, affordable tools. All you're going to need is a transparency sheet, a fine-tipped marker, scissors, scale or a ruler, and of course a microscope. To make your measurements more accurate, it would be helpful, though not essential, to have a stage micrometer to calibrate your setup. To measure cells under a microscope, we need a reference scale. That's where the eyepiece graticule and the stage micrometer come in. Eyepiece graticule is a pattern or a set of markings placed in the focal plane of a microscope to provide a reference point or aid in measurement. It's also called eyepiece reticle or eyepiece micrometer. A stage micrometer is a slide with a precisely measured scale on its surface. Here's the basic idea. The objective lens magnifies the specimen first, forming an intermediate image at the focal plane inside the eyepiece tube, right where the eyepiece diaphragm is located. If we place a transparent scale in this diaphragm, the scale appears superimposed over the image of the specimen. Then, the eyepiece lens magnifies both the image and the scale together, creating a final view with a built-in measurement tool. Commercial eyepiece graticules are available widely, but they are often costly. Instead of buying those, we'll make our own. It's budget-friendly and surprisingly fun. So first, I measured the inner diameter of my eyepiece, which is about 2 centimeters. I can also see that both the lenses of the eyepieces are less than a centimeter long. I also checked the length of 10x and 15x eyepieces. Then I cut a strip of transparency about 1.5 cm wide and 5 to 6 centimeters long. At about 7 mm from one end of the strip, I marked 10 dots, each 1 mm apart. Here is the design of the graticule I made. I folded the strip at 1.5 centimeters from the base and kept the holding part at 8 millimeters so it could sit snugly inside the eyepiece. You can adjust the length and the width based on your eyepiece measurements. And finally, I inserted the strip near the field lens of the eyepiece. If you have access to an advanced microscope, it is likely to have a graticule holder installed just outside the field lens, so check with the manufacturer. As you can see, I can rotate the graticule for easy measurements. And here is how a fishing line or a braided thread looks like with a built-in reference graticule. I've used light from above. Now let's try to understand the measurements. So what does one division on this graticule mean when seen under the microscope? So the idea is the objective lens magnifies the specimen. The graticule placed at the eyepiece is not magnified by the objective but only by the eyepiece lens. In other words, when total magnification is 100x, graticule magnification is 10x. So under 10x objective and 10x eyepiece, one division of the graticule corresponds to 0.1 millimeters on the specimen. If you switch to a 45x objective, the specimen is magnified even more, but the graticule stays the same. So now, one graticule division is equal to 0.022 millimeters. Let's quickly measure thickness of hair with the graticule now. Under 100x, hair width would be 0.1 millimeter. Under 450x, hair width is calculated to be 0.08 millimeters. Of course, this is a theoretical value, so it assumes perfect scaling and alignment. For accurate results, we'll have to calibrate using a stage micrometer. So to confirm our measurements, I've used a printed stage micrometer, and the least count of this is 0.2 millimeters. To calibrate, I placed the stage micrometer instead of slide on the stage, and I took images of graticule with the stage micrometer. If you don't have a stage micrometer, you can think of placing a graph paper and lights from top. It should certainly work up to 100x or 150x. So here's what I found. Under 10x objective, for both the eyepiece combinations, I expected 10 graticule divisions to align with 1 millimeter. Instead, 8 graticule divisions aligned with 1 millimeter. Therefore, one division is equal to 0.125 millimeters. Under 40x objective, for both eyepiece combinations, I expected 8 graticule divisions to align with 0.2 millimeters. Instead, 6 graticule divisions matched 0.2 millimeters, so one division is equal to 0.033 millimeters. I was disappointed and I made 3 graticules, just to be sure, but got similar calibration every time. So the 10x objective of microscope is perhaps magnifying 8x and the 45x objective is giving effective magnification of 30x. Now, using our more accurate estimate, width of the hair is calculated to be 0.125 millimeters under 10x or 0.115 millimeters under 45x. Here are a few quick tips before you build your own eyepiece graticule. So commercial tools are more precise and they are ideal for advanced scientific work. But your DIY graticule, especially if calibrated with a stage micrometer, gives reliable estimates too, and it's perfect for school labs, home learning, and early research. Even if you don't have a micrometer, your graticule alone is still a nice tool for exploring relative cell sizes. Most high schools and colleges in India use a microscope similar to what we are using in this course. So check with the manufacturer for exact location of graticule holder if you have access to a more advanced microscope. Try it out and share what you measure. In the next video, we'll study osmosis in Tradescantia leaves. Thank you so much for joining and happy scaling.